continue with showing images and talking about ependymomas. Ependymomas are tumors derived from the ependymal lining of the ventricles, and they can also occur in the central canal of the spinal cord. Because this is brain bit by bit, I'm only going to talk about the intracranial ependymomas. 2/3 of intracranial ependymomas occur in the posterior fossa in the fourth ventricle, and ependymomas are very soft tumors accommodating its form and extruding through the foramina of Magendie and Luschke, and ependymomas are sometimes called a plastic tumor. The presenting complaints are related to the CSF obstruction, and patients present with hydrocephalus. The supratentorial ependymomas are often located extraventricular, thought to be derived from remnants or nests of ependymal cells. And supratentorial ependymomas are partly cystic and partly solid. About half of them have calcifications. They're typically quite large at presentation, and if they are located near the cortex, they can present with seizures. Ependymomas can spread through the CSF, so you always have to do imaging of the entire neuraxis if you think of an ependymoma to check for leptomeningeal metastasis. The ependymomas from different anatomical regions are histologically identical, but in the different anatomical regions they have very distinct biological and clinical behavior, and that's why in the WHO classification from 2021 you can see that the ependymomas have been classified by compartment, so supratentorial, posterior fossa and spinal, and at each compartment you have different gene mutations and different molecular markers, and in different age group there is a predominance of a certain gene mutation. And these molecular markers and these different tumors also have different imaging features and different prognosis and treatment options. So nowadays, you might see an ependymoma being described as a ZFTA fusion positive supratentorial ependymoma, partly solid, partly cystic, with the cystic part being hyperintense on FLAIR images. And although the molecular markers are important, the neuropathologist in our hospital always says you have to make diagnosis of brain tumors on your basic histology, so on your pink and purple H&E staining. And ependymomas are characterized by these pseudorosettes, where the tumor cells are arranged radially around a blood vessel. And the ependymal cells are derived from radial glial cells. I talked a lot about radial glial cells in the first videos on this channel. So the radial glial cells, some of them become neural stem cells and many of them divide and become ependymal cells. Ependymal cells are not neural stem cells anymore, and these ependymal cells, they have these cilia, and these cilia are important to influence the CSF flow, and the ependymal cells itself form a barrier between the CSF and the extracellular fluid of the brain parenchyma. And we are going to come back to that when we will talk about the glymphatic system. These are images from human stillborn fetuses. This is a 24-week-old fetus where you can see the ependymal lining with the cilia on the ependymal cells. And you can see a radial glial cell located between the ependymal cells with the cell body in the ventricular zone, and then the process with the migrating neuron going towards the cortical plate. And you might remember that the basal process of the radial glial cell went all the way to the external glial limiting membrane. And there's a subtype of ependymal cells that's called a tannicyte, occurring mainly in the third ventricle that also has a long basal process going all the way up to the external glial limiting membrane. And these tanycytes are a subtype of ependymal cells, and you can see in this 24-week-old stillborn fetus that the tanycyte radiates towards a blood vessel. And when I was looking at the relation between the radial glial cells and the neural stem cells and the ependymal cells, I came across an interesting article describing how the ependymal cells become arranged in pinwheel structures postnatal. So these are postnatal images, so P5 and P7 are different types postnatal. And the radial organization of the ependymal cells, and you can see this is a staining, the red one where the cell membrane has been outlined, that all the ependymal cells have this pinwheel arrangement, and that resembles a little bit the pseudorosettes of the ependymoma. The ependymal lining is derived from the radial glial cells lining the neural tube, but there's also part of the covering of the primitive ventricles that becomes choroid plexus, and you can have tumors arising from the choroid plexus as well, and we're going to look into that in the next video before we go to the lymphatic system.